Associated glands are present outside the alimentary tract and are connected to it by ducts. Associated glands include salivary glands, which may be either accessory salivary glands present in the oral cavity as labial, buccal, lingual, palatine, and tonsillar, or major salivary glands, which are either serous glands as the parotid or mixed mucoserous glands as the submandibular and sublingual glands. Other associated glands important in the digestive system are mixed in function, both exocrine and endocrine, as the pancreas and the liver. Associated glands are formed of connective tissue stroma and parenchyma. Stroma is made of capsule trabeculae dividing the gland into lobes and lobules and reticular stroma. Parenchyma consists of ducts carrying secretion to different sites of the body and cells arranged in cols or acini for forming the different types of secretion.
Liver. It is the largest organ in the body and is both an exocrine gland secreting bile into a duct system and an endocrine gland releasing several substances into bloodstream. It is formed of stroma and parenchyma. Stroma of the liver is composed of capsule, trabeculae, reticular stroma, and portal canals. Capsule. Capsule of Glassons is thin and made of loose connective tissue covered by peritoneum. It is thickened at the porta hepatis. Porta hepatis is a depression in the undersurface of the liver and is the site of entrance of blood vessels and exit of hepatic ducts and lymphatics. Trabeculae are also loose connective tissue extending from the porta hepatis, dividing the liver into lobes, right and left lobes, and lobules. Lobulation is not clear in human, but it is clear in certain animals as pig. It became clear in human in certain diseases as liver cirrhosis. A reticular stroma is formed of radially arranged reticular fibers supporting the liver cells, extending from the periphery towards the center. It is thickened around the central vein and can be demonstrated by silver stain. Portal canal is a triangular mass of connective tissue found at some corners of hepatic lobules. Not all. It contains a branch of hepatic artery, a branch of portal vein, a bile duct, and a lymph vessel. Parenchyma of the liver can be defined by three ways: classic hepatic lobules, portal lobules, and liver acini. Classic hepatic lobule. It is a hexagonal mass of liver tissue drained by a central vein. Portal lobule. It is a triangular mass of liver tissue formed of bars of three adjacent hepatic lobules and is drained in one bile duct. Liver acinus. It is considered as the structural and functional unit of the liver. It is a diamond-shaped mass formed of a triangular path of two adjacent hepatic lobules and is supplied by a terminal branch of the portal vein, a terminal branch of hepatic artery, and is drained by a terminal branch of bile duct.
Classic hepatic lobule is a hexagonal mass of liver tissue drained by a central vein. It consists of radially arranged cores or plates of liver cells, hepatocytes, supported by delicate stroma of reticular fibers, and separated by radially arranged blood sinusoids. Each hepatic plate consists of 2 or more rows of hepatocytes. Bile canaliculi are present between the rows of hepatocytes. Hepatocytes are polyhedral in shape with central round vesicular nuclei and may be binucleated. The cytoplasm contains organelles and inclusions. Organelles are detected by electron microscope. They are numerous mitochondria, Golgi body, ribosomes, lysosomes, peroxisomes, smooth endoplasmic reticulum, and rough endoplasmic reticulum. Inclusions are glycogen. It can be demonstrated by Bess-Skarmin. In ordinary sections, liver cells or hepatocytes appear vacuolated as a result of dissolved glycogen. Fat is also present in liver cells but is masked, and it is found in combination with proteins. It becomes visible in fatty degeneration of the liver. By electron microscope, long microvilli project from the surface of hepatocytes facing blood sinusoids, while short microvilli project from the surface facing bile canalicule. Blood sinusoids are lined partially by fenestrated endothelial cells and partially by von Kappa cells. Blood sinusoids are demonstrated by gelatin-carmine injection. Von Kappa cells are large branched phagocytic cells derived from monocytes lining liver blood sinusoids. They are rich in lysosomes and can be stained by vital stains as trypan blue. They play a role in destruction of senile RBCs and formation of bile pigments. Space of this is the space that separates hepatocytes from the walls of blood sinusoids. It is also called perisinusoidal space. It contains microvilli projecting from the surface of hepatocytes, plasma, reticular fibers, lipocytes, which are fat-containing cells, and non-myelinated nerve fibers. Its main function is the exchange of materials between bloodstream and hepatocytes which do not contact the bloodstream. Bile canaliculi, on the other hand, are an expansion of the intercellular space. They have no special lining epithelium, but only the adjacent plasma lemma of the two neighboring hepatocytes.
Space of this structure. Space of this is a narrow space, separates hepatocytes from the walls of blood sinusoids. It is perisinusoidal space. Contents. Space of this contains microvilli projecting from the surface of hepatocytes, plasma, reticular fibers, and lipocytes, fat-containing cells, and non-myelinated nerve fibers. Function of space of this. Its main function is exchange of materials between bloodstream and the hepatocytes which do not contact the bloodstream. Second important function is to prevent collapse of blood sinusoids due to its content, microvilli projecting from hepatocytes into the space of this, Adipocytes are considered supporting cells. They store beside fat vitamin A. Reticular fibers traversing the space, forming a support for the wall. The hydrostatic pressure in the sinusoid is equal to that of the space of death, as both contains plasma. Microfilaments and microtubules present in the endothelial cells act as cytoskeleton.
Portal lobule is a triangular mass of liver tissue whose three apices are neighboring central veins. It consists of bars of three adjacent hepatic lobules. It has the portal area in its center, formed of masses of connective tissue found at the corners of three hepatic lobules. It contains an artery, branch of hepatic artery, a vein, branch of portal vein, a bile duct, and a lymph vessel. Hepatocytes of the portal lobule are drained into a single bile duct present in the portal tract. Liver acinus is considered the structural and functional unit of the liver. It is defined as the mass of liver tissue that is supplied by a terminal branch of the portal vein, hepatic artery, and drained by a terminal branch of the bile duct. It is a diamond-shaped mass of liver tissue formed of triangular bars of two adjacent hepatic lobules. The bases of the two triangles parallel each other, and the apices include the central vein and the two adjoining portal areas. The liver acinus is surrounding a central vascular core containing branches of hepatic artery and portal vein. This explains why the peripheral hepatocytes are richly supplied with blood, while the central hepatocytes are poorly supplied with blood.
Liver acinus. According to blood supply, the acinus is subdivided into three zones that correspond to distance from the arterial blood supply. Zone 1 contains hepatocytes closest to the arterioles. They are best oxygenated and best nourished. Zone 2, those hepatocytes intermediate between Zone 1 and Zone 2. These hepatocytes have poorer blood supply and nourishment than Zone 1. Zone 3, those hepatocytes furthest from the core have the poorest blood supply and nourishment. Clinical concept of liver acinus. In toxemia, the presence of toxins in the blood, Zone 1 is the most affected zone, as it is nearer to the blood supply. In hypoxemia, hepatocyte necrosis is more encountered in Zone 3, centrilobular.
Bile secretion or drainage. Bile is secreted into the bile canaliculi found between rows of hepatocytes. Bile canaliculi have no special lining epithelium, but only the adjacent plasma lemma of the two neighboring hepatocytes. Then bile passes to the canals of Herring, found at the periphery of classic lobules. Canals of herring are lined by simple squamous epithelium. Then bile passes to small bile ducts lined with simple cubical epithelium. Then to large bile ducts lined with simple columnar epithelium. These large bile ducts join to form two large extrahepatic ducts, the right and the left, which in turn unite to form the common hepatic duct. Common hepatic duct is lined with columnar epithelium. It is joined by the cystic duct coming from the gall bladder. Cystic duct is also lined with simple columnar epithelium. When it joins the common hepatic duct, it forms the common bile duct. Common bile duct is lined with tall columnar cells and delivers its content into the lumen of the duodenum at the duodenal papilla ampulla of water. When common bile duct is blocked, bile accumulates in the liver and the blood, causing yellowish coloration of the skin, a condition which is called jaundice. Bile secretion or drainage. Bile is secreted into the bile canaliculi found between rows of hepatocytes. Bile canaliculi have no special lining epithelium, but only the adjacent plasma lemma of the two neighboring hepatocytes. Then bile passes to the canals of herring, found at the periphery of classic lobules. Canals of herring are lined by simple squamous epithelium. Then bile passes to small bile ducts lined with simple cubical epithelium. Then to large bile ducts lined with simple columnar epithelium. These large bile ducts join to form two large extrahepatic ducts, the right and the left, which in turn unite to form the common hepatic duct. Common hepatic duct is lined with columnar epithelium. It is joined by the cystic duct coming from the gall bladder. Cystic duct is also lined with simple columnar epithelium. When it joins the common hepatic duct, it forms the common bile duct. Common bile duct is lined with tall columnar cells and delivers its content into the lumen of the duodenum at the duodenal papilla ampulla of water. When common bile duct is blocked, bile accumulates in the liver and the blood, causing yellowish coloration of the skin, a condition which is called jaundice. Bile secretion or drainage. Bile is secreted into the bile canaliculi found between rows of hepatocytes. Bile canaliculi have no special lining epithelium, but only the adjacent plasma lemma of the two neighboring hepatocytes. Then bile passes to the canals of herring, found at the periphery of classic lobules. Canals of herring are lined by simple squamous epithelium. Then bile passes to small bile ducts lined with simple cubical epithelium. Then to large bile ducts lined with simple columnar epithelium. These large bile ducts join to form two large extrahepatic ducts, the right and the left, which in turn unite to form the common hepatic duct. Common hepatic duct is lined with columnar epithelium. It is joined by the cystic duct coming from the gallbladder. Cystic duct is also lined with simple columnar epithelium. When it joins the common hepatic duct, it forms the common bile duct. Common bile duct is lined with tall columnar cells and delivers its content into the lumen of the duodenum at the duodenal papilla ampulla of water. When common bile duct is blocked, bile accumulates in the liver and the blood, causing yellowish coloration of the skin, a condition which is called jaundice.
Gallbladder. It is a hollow, pill-shaped organ attached to the lower surface of the liver. Its wall is made of mucosa formed of epithelium and chorion, musculosa, and outer fibrous coat or serosa. Mucosa. It is highly folded, especially when the gallbladder is empty, and is formed of simple columnar epithelium, partially secretory and partially absorptive cells having apical microvilli. The epithelium is resting on a chorion containing mucous tubuloalveolar glands near the neck of the gallbladder. There is no submucosa. Musculosa is thin and consists of irregularly arranged smooth muscle fibers with some collagen and elastic fibers. Adventitia or serosa. It is formed of loose connective tissue containing blood vessels and nerves covered by mesothelium, simple squamous epithelium. Serosa covers most of the gallbladder, except where it is attached to the liver. Function of gallbladder. Concentration and storage of bile through absorption of the excess of water. Evacuation of the bile into the duodenum. Secretion of mucus by the epithelial cells of the gallbladder.
Blood supply of the liver comes from hepatic artery, which carries oxygenated blood, and portal vein, which carries absorbed materials from the gastrointestinal tract, and hemoglobin breakdown products from the spleen. Both hepatic artery and portal vein power their blood into blood sinusoids. Then blood passes from blood sinusoids and reaches the central veins, then passes to sublobular veins, then to tributaries of hepatic veins. Finally, the venous blood is drained into inferior vena cava. Functions of the liver. The liver has an exocrine as well as an endocrine function. Exocrine function of the liver is the production of bile, which is a product of hemoglobin breakdown. Bile contains bile acids, salts, pigments, cholesterol, and neutral fats. Bile salts are important in the breakdown and digestion of fat. Endocrine function is the release of glucose and plasma protein, prothrombin, fibrinogen, albumin, and so on. Liver also has important other functions, like storage functions, storage of carbohydrates in the form of glycogen, and maintenance of normal glucose blood level. Also storage of vitamins A, B, D, and K. Storage of minerals like iron and copper. glyconeogenesis, formation of glucose from amino acids and lipids, detoxification of drugs and hormones, phagocytosis of snail RBCs by von Kupffer cells,